Hello, we're going to be looking at the spectrophotometric determination of alcohol content of beer and mouthwash. And this is effectively the lab scale version of a breathalyzer test. And of course, the point of a breathalyzer test and the point of this particular lab is to detect and quantify the amount of alcohol that's in a sample. Now, our samples are going to be liquid samples or aqueous samples, and we're going to be detecting the alcohol content by a change in color and measuring the intensity of that absorption of that particular color. So let's talk about this some. The reaction that we're actually going to be looking at is down here at the bottom of the first page, and it takes dichromate, which you've seen me use before, dichromate, and add some acid to it, and then, important part here, add alcohol to it. And this alcohol reduces in an acidic solution, causes the reduction of chromium from chromium 6+, which it is when it's in this dichromate form, to chromium 3+. Now, the thing that really allows us to figure out what's going on here is that dichromate is a yellow-orange solution. Alcohol, of course, is colorless, as is the acid that we're going to be using. But when it's reduced, when the chromium is reduced from 6+ to 3+, it becomes a blue-green color. So it also makes some acetic acid and water. So what we're going to try to do today is determine the amount of blue-green that is made directly proportional to the alcohol. That's the whole point of this. But we're going to have to detect it in a yellow-orange solution, and that's going to be a challenge. Why are we going to detect it in a yellow-orange solution? Because this is going to have to be in excess. We want to detect the amount of alcohol, so this has to be the limiting reactant. Okay? So we're going to have excess dichromate, therefore there's going to be some orange-yellow solution still, and we're going to be making blue in it. So it's going to become a brown solution upon the creation of that chromium 3+. These two colors are going to form a brown solution. So quantifying how much blue is in brown is not something we can do just with our eyes. So let's come up here and talk just a little bit about electromagnetic radiation, because we know we're going to have to be detecting a color. How are we going to do that? Well, as we know, visible light is somewhere between 400 and 700 nanometers, specifically between 380 and 750 nanometers. Now, you can have polychromatic light, which is what we're going to see in our solution, polychromatic light, which has a multitude of wavelengths. Now, truly, completely polychromatic light looks white to us, okay, or colorless. What we're going to be looking for is not a group of wavelengths, is not white. We're going to be looking for that blue. So the first thing we're going to be doing is looking for that one wavelength, that monochromatic light that allows us to have the maximum absorbance for chromium 3+ and the minimum absorbance of the dichromate, of the yellow-orange. Trying to separate those out, which our eyes can't do, but a spectrophotometer can. A spectrophotometer can observe a single wavelength, and that's what we're going to be doing. Now, understand that we're going to be seeing a color that's complementary to the one that's being absorbed. When we see a green-blue solution, like chromium 3 plus would be if it were all by itself, the color that's actually being absorbed by the spectrophotometer is orange. And when we're looking at the dichromate solution, which is yellow-orange, the color that is actually being absorbed is in the blue-green region. So that's kind of cool. We have to remember that we see the complementary color that is from that which is being absorbed. Okay, so let's go on with this. We have decided then that alcohol can be detected because they are a direct proportion. The amount of alcohol is directly related to the chromium 3 plus. So we can detect the amount of alcohol by measuring the amount of chromium 3 plus, and we're going to do that by monitoring a single wavelength that gets the maximum absorbance of chromium 3 plus and the minimum absorbance of dichromate. Okay, so here we go. Here we have a graph, and this graph is an absorption spectrum. What's an absorption spectrum? Well, this is a graph essentially of the absorbance versus the wavelength for a particular species. Now, as we look at this, we're going to see the dichromate is the one with the open dot. So the dichromate's absorption spectrum, it's around 2.7 at 400 nanometers, 400 to 460, and then it drops significantly and steadies out around 2, and then it drops some more. So that by the time it's around 540, 560 nanometers, there's very little absorbance at all. On the contrary, we've got our chromium 3 plus, which also absorbs significantly in those lower wavelength sections, but has a regional maximum right around, not the way I drew it, but right around 580 nanometers. Now that's very significant because once again, we are trying to find the wavelength where we get the maximum absorption of chromium 3 plus at the same place we get the minimum absorption of the interference or dichromate. And it would appear that that's going to be right around 580 nanometers. Sure, we'd get a maximum absorption of chromium 3 plus down here in the 400 range, but we'd have so much interference from the dichromate we wouldn't be able to distinguish them. So 580 nanometers is what we're going to set our spectrophotometer to. How does it allow us to figure out the concentration? Because that is, after all, the point of this experiment. Well, we can figure it out using something called the Beer-Lambert law. And the Beer-Lambert law is simply absorbance equals eta, and eta is called the molar absorptivity, and that's a constant for a particular absorbing species and that particular wavelength at which you're measuring it. L is the path length. Path length of what? And finally, C is the concentration. So you can see here that absorbance is directly related to the concentration. If you only know molar absorptivity and path length. Now this path length, what am I talking about? Well, over here we have a detector. Okay, I'm going to write the worst version of a spectrophotometer you've ever seen. And here in the middle is our solution. And the width of this particular vessel in which we put our solution is 1 centimeter. as exactly as we can get it. Over here, we have the source of in our case, it's going to be 580 nanometers of monochromatic light. Now, some of this will actually be absorbed by the solution. So maybe one gets through, or maybe two out of a whole bunch of wavelengths that are being shot at the solution. That measures the transmittance. We can measure the amount that's absorbed by the difference between the amount of light that was sent out and the amount that was detected. Okay, so that's where our path length came from, and that's really important. And all of our cells that we're going to be using in this experiment are exactly 1 centimeter. Okay, so good. Now we know that we can use the Beer-Lambert law to find concentrations concentrations from absorbances. That's great, but I don't know the molar absorptivity. How do I figure that out? Because that's certainly important. We've got this, we know this, we want this, and we've got this molar absorptivity that's an unknown. Well, we're going to get around that. And the way we get around that is by making a number of standard solutions. Solutions that have a known amount of alcohol, a known percentage of alcohol that you're actually going to make. And then you're going to measure the absorbance for those known samples. They're going to be made exactly the same way that your unknown is made. So the same solution, so that in theory, they're all going to have the same molar absorptivity. So we knock that right out. So what we're left with is a group of samples that we know the concentration of alcohol, and we can measure at 580 nanometers the absorbance, and then we can plot it. And it should, if you've done it correctly, plot a nice straight line. So we can see that we would need to plot absorbance versus concentration. And that would give us our molar absorptivity. So here we go. We've plotted percent alcohol. Let's see, I guess we made up a 2.5% solution of alcohol, and we ended up with an absorbance of 0.2. And then we made up a 4.5%, and we got an absorbance of 0.35, etc. We plotted it using LoggerPro, and we got an equation of a line. And it told us what the slope was, and that's going to be very important because that's eta, and it tells us what our error is. Okay, so great. What's the point of this? Well, the point of this now is you're going to make some unknowns and measure their absorbance, and say one comes out at 0.6. Our percent alcohol would be 9. Obviously more accurately than that. If we had one that was 0.7, right across to here, that would be a 10.5% alcohol. See, so that's what we're going to do. First part of the experiment, we're going to make a standard curve using known samples, known percentages. And from that, we're going to measure the absorbance and plot to form the standard curve. Then again, we will take the unknowns and the standard curve equation of a line, and we'll find the percent alcohol in the unknowns. Now all of this down here is explaining how we get absorbance from the percent transmittance. Because our old spectrophotometers gave us percent transmittance, we had to convert it to absorbance. But these that we're going to be using are going to take absorbance for us.